In this video, I'm going to show you a diagram of a transfer RNA molecule. So a transfer RNA molecule is what brings the amino acids into the ribosome to be added to a growing polypeptide. And it is made up of a series of loop and hairpin domains. And when we look at it kind of smushed flat in 2 dimensions, it looks kind of like a clover leaf with 4 different arms. So here we've got our what's called the D arm, sometimes called the DHU arm. Here we've got the anticodon arm. With our anticodon being 3 nucleotides right there. We've got a structure called the variable arm. We've got what's known as the T psi C arm. And then we've got the amino acid acceptor arm. And should note that this acceptor arm, where the amino acid is charged onto the transfer RNA, is always going to end in the sequence CCA. These are ribose nucleotides, of course. And it's at that 3' end that the proper amino acid is going to be added by aminoacyl-tRNA synthetase, the proper aminoacyl-tRNA synthetase that binds, that recognizes the anticodon. So if we have, for example, our anticodon CCA, for example, that's going to be add proline up here at the acceptor arm. Or CC anything really, because of wobble, CCA, CCG, CCU, CCC, all of those are going to encode proline. Okay, so another thing to note is that when our transfer RNA is charged by the enzyme aminoacyl-tRNA synthetase, it is done in a way that it consumes ATP and when the ATP is consumed, it's going to release pyrophosphate. So that means we're going to have a very high energy bond between that adenine at the end there and that amino acid. And the significance of that is that that's going to provide the energy to form the peptide bond once this transfer RNA gets into the ribosome. So it's kind of giving it a source of energy that's going to be spent on forming that peptide bond in during the actual translation.